Sí, no, pero ese se oye ahí solo.
Vale. Vale. Hace 2 años yo decidió elegirnos a nosotros para poder hacer el trabajo fin de máster de un tema un poco complejo porque era de leptoconos, un insecto que casi nadie conocía. Y bueno, aún así tuvo la valentía de decidir escogernos y hizo el trabajo fin de máster, lo defendió de forma brillante y consiguió hace poco una beca para hacer la tesis doctoral con Jordi Figuerola y conmigo. Y nada, vamos a escucharle a ver qué nos cuenta sobre estos bichos. Muchas gracias.
Thank you very much. Hi, my name is Ionut. As Sergio said, I have started my PhD this year, and the last year, in the mosquito Figuerola lab, under the supervision of Jordi Figuerola and Sergio Magallanes. And today I'm going to talk about my PhD plan, which is titled Ecology of Leptoconops midges and sandflies in Andalusia. I've been working on it for the past 10 months. So far, regarding my academic background, I completed a degree in biology at the University of Sevilla. Then I completed a master's degree in Biodiversity and Conservation Biology at the University of Pablo de Olavide. Then I do a master in teaching. And finally, in April of 2025, I started my PhD here at Estación Biológica de Doñana, thanks to Junta de Andalucía predoctoral grant. To begin with, when we conduct a systematic literature research on Web of Science, using the keywords of mosquito, mosquito, ecology, parasite and disease, we can retrieve more than 30,000 publications about that topic, which indicates that this topic is well researched due to the fact that mosquitoes play an important role in the diseases transmissions, mostly to humans. When we do the same using BT midges, we observe that the number of publications are lower, and that's because they didn't play the same role as mosquito in the transmission of diseases to human. However, when we do a more specific search with one of the topic of my PhD on Leptoconops, the number of publications are only 62, which shows us that the gap of knowledge of this is notorious and nothing is known about the ecology and the parasite transmission for this genre. Parallelly, when we do for sandflies, which is the other topic, the same pattern, the same number of publications can be retrieved from the search, with fewer and more publications than biting midges, just because sandflies are the main vector of leishmaniasis, which is a disease transmitted to humans, so there is that importance. Vector-borne diseases have become a major health concern in the recent years, along with the One Health initiative. These diseases have been transmitted by different blood-sucking insects, such as mosquito, biting midge, or sandflies, or ticks. These act as vector. They eventually can bite some infected vertebrate host and then carry that parasite and develop it. And then when they bite another vertebrate, which is uninfected, they can transmit that parasite. If the transmission is successfully done, that vector is called competent for the transmission of that disease. When the transmission is made from animal host to human host, it calls zoonotic transmission, and between animal host is called sylvatic. A global change driver have a positive impact on vector-borne diseases. One of the driving forces is climate change, where rising temperature, prolonged drought, and also extreme weather events can create a suitable condition for the suitability of vectors and the expansion, permit the expansion of these vectors. On the other hand, habitat human activities such as land use change, habitat fragmentation, urbanization leads to a disruption in the dynamics of vector, parasite, and host. So this permits the prevalence of blood-sucking insects to increase in urban area, and therefore the contact between humans and vector increases. Furthermore, human travel and global trade leads to facilitation of the dispersal of both vector and pathogen across borders. And finally, agricultural intensification and the increase in livestock density creates suitable environments for the vector population increase. And this leads into more opportunities of diseases transmission. My research is focused on two groups of less studied than mosquitoes, which are hematophagous. These groups are biting midges from the family Ceratopogonidae. But specifically, I will be focused on Leptoconops midges. And the other topic is sandflies from the family Psychodidae. Now we're going to talk about some of the characteristics of their biology. Both of them are a widespread distribution, absent from the poles and remote islands. Additionally, sandflies are also absent in cold regions at high altitude. Both display hematophagous habits, where the female adult depends on blood intake for the egg development. In case of the Ceratopogonidae family, 4 genera are hematophagous, and in case of sandflies, all genera from the subfamily Phlebotomine display this habit. In case of the active period, Leptoconops are diurnal, being more active during the day, the first hour of the day, while sandflies are more active during the dusk and dawn, displaying a crepuscular habit. And finally, both of this group are implicated in the transmission of several diseases. In case of biting midges, they can transmit some viruses for the orbivirus complex, such as blue tongue virus to livestock, and parasite from the Proteus and Trypanosoma genera. And in case of sandflies, they can transmit parasite from Leishmania genera to human and animals, and viruses from the Papataceae fever complex, such as Granada and Toscana virus. When we go to GBIF to observe the number of occurrences of the Leptoconops midges, we observe that only few occurrences can be retrieved from that search. And most of them, these occurrences are from fossil species. So therefore, nothing is known about the distribution of the species that are nowadays in Europe. When we do with sandflies, much more number of occurrences can be retrieved, and most of the distribution of that occurrence are in the southern region, in the Mediterranean area, which displays the suitable condition from the species survival and development, whereas the northern region didn't display that condition, so there is no presence of this species. Both groups display a similar life cycle. We start when a gravid adult female oviposit the eggs on organic substrate. That eggs hatches and then develop into larvae, passing through 4 stages that can last from week to month, and then they develop into pupa. That pupa emerges into adult female and males, and they start to feed on sugar to meet their energetic requirement, and while they start mating. After mating, a female adult may feed on vertebrate blood to complete the egg development. In this stage, they may take successive blood intakes, so they may occur different transmission of parasites between the hosts. And then when the eggs are developed in the abdomen, the gravid females again lay that eggs, and the cycle starts again. With this in mind, what do you know about Leptoconops? In Spain, only 4 species have been documented. One of the species is Leptoconops irritans, Leptoconops noei, Leptoconops kertetsi, and Leptoconops bethi. However, nothing is known about the ecological factors that determine these different species' survival and development in Spain, and nothing is in that region, and nothing is known about the vectorial role of this genera, which is hematophagus. So with this, we identify the following knowledge gaps: species diversity, geographical distribution, and also the phenology of these species in Andalusia, the ecological factor that permits a species survival and development, the vectorial role of the genus, and the feeding patterns of the genus. And what is known about sandflies? Much more is known about sandflies due to the implication in the transmission of leishmania, which is the main vector. The distribution is well known all across European regions due to the implication in human health concerns. Presence, as I said, is in the southern regions, which are in red, and in green are the absence of the different species. These 4 maps are from species that act as vector of leishmania in Europe. So the distribution is known due to the fact that some institutions, European institutions, such as the European Center for Disease Prevention and Control, monitor periodically, and some research initiatives such as CLIMOS project. In the Mediterranean, leishmaniasis is endemic, and in Spain as well. And in case of Spain, the incidence rates of the infection are from 0.8 to 0.6 in the recent years, cases per 100,000 humans. And also in recent years, between 2023 and 2025, annually have been recorded more than 13 to 1,400 cases, human cases of leishmaniasis. Also in Spain, 2 major outbreaks of leishmaniasis have been registered. One of them in Fuenlabrada, Madrid, during 2009-2012 period. During that period, more than 500 cases of human cases have been recorded. The cause of these outbreaks was the unusual sylvatic cycle, where leporids, such as hare and rabbit, play as main reservoir of leishmania parasites, and also the land use change from cropland to parks, which permit the sandflies to have new breeding sites near housing, so the contact between humans and sandfly species were more intimate. The second outbreaks were registered in Córdoba during 2023-2024 period. In Córdoba main capital, and also in some towns nearby. In that period, the cases rises from 7 to 31. As in Fuenlabrada, the cause was the implication of leporids as main reservoir of the transmission. With that, what do we identify as knowledge gaps for DIPS? One of the knowledge gaps are which are the blood feeding pattern of the different species of sandflies, and which is the risk of leishmania transmission in Andalusia. So, therefore, the main aim of my thesis is to unravel the ecology and the biology of these 2 groups. For that, my thesis will have 2 main sections with 3 chapters. The objective of the section 1, the first one is to determine the species composition of Leptoconops species in Andalusia. The second, to elucidate the phylogenetic position of the genera within the Ceratopogonidae family. The third one, to determine the ecological factor that permit the species development through habitat suitability modeling. And the fourth, to understand the blood feeding patterns and to detect parasites. This will be composed of 4 chapters. And the section 2, the objective of the section 2, will be to revise the blood feeding patterns of the sandfly species in Europe and to identify the area with high risk of transmission of leishmaniasis in Andalusia. Regarding the methods, the study area is Andalusia, which have been sampled through 2023 and 2024 period. 951 points were sampled 3 times per year, 1 season. Thanks to our BioPrevent project, were sampled in Huelva, Seville, and Cádiz. And in 2025, were sampled around 194 points all across Andalusia, sampled weekly from May to November, thanks to Junta de Andalucía Vector Surveillance Project. The sampling methodology used was the BG Sentinel traps baited with CO2. The capture was stored in the cryofreezer and then morphologically identified to species level. These parts, the field work and all the identification were made by our technical team. For the first chapter, the aim is to determine the species composition of Leptoconops. In the Western Palearctic, there have been documented 34 species of Leptoconops, most of them in the northern African region, some in Italy, France, and Russia. For this study, we used the capture from the 2023 and 2024 sampling period. The Leptoconops midges were first sorted into different groups using observable characteristics under the stereo microscope. We form different groups. From each group, we select around 10 specimens to mount them in using Hoyer mounting medium. The slide-mounted specimens then were identified to species level using different Western Palearctic fauna keys. Some species could be identified to species level using that key. However, some species didn't be possible to identify due to morphological differences. So we determined that species were new. We do the description of the different species and also using the correct method, the correct terminology, following different Diptera manual. We deposited the holotype and some paratype in entomological collection here at Estación Biológica Doñana. As well, we sent some paratype to the Museo Nacional de Ciencias Naturales in Madrid and in the National History Museum in London. Then, to confirm the morphological identification, we also do molecular identification analysis using DNA barcoding. We sequence the COX-1 gene of each species, and with that sequences and the sequences that we retrieve from Leptoconops species from gene bank, we construct a phylogenetic tree using a maximum likelihood approach. The species that we recorded, the new ones were Leptoconops nigrithorax, Leptoconops triangularis, Pseudoirritans, and communis. And the already described species that we also recorded in our study was Leptoconops irritans, Videntatus, Noe, and Kertesi. In case of Leptoconops videntatus, is a new record from Spain also. Now I'll talk you about the characteristics of the new species. Leptoconops nigrithora is characterized by a shiny black thorax and entirely black-brown legs. Leptoconops triangularis characterized by triangular pattern on tergites. And Leptoconops pseudoirritans is similar to Leptoconops irritans. That's the name, false irritans. However, have some difference, such as is the length of the body is shorter, and some characteristics of the palpus are also different. Leptoconops communis is similar to Leptoconops besi, however, displays some differences in the number of sensory pits. The hyaline sensory setae is have different shape, and also the humeral pits are different. When we assess the phylogenetic relationship of the species, we determine that the sequences of our species cluster all together in monophyletic clades in different each species, and the sequences of the species that we retrieve from gene bank in other clades all the clades have a high bootstrap value, so this could confirm the morphological identification that was done well, and the species that were new is clearly new. This article is now under review in Sukis. For the second chapter, we aim to determine the phylogenetic relationship of the genus within the Ceratopomyidae family. For that, we will sequence 3 genes that have been sequenced from Culicoides species, which is the sister taxon of Leptoconops, with all the sequences of our new species, from our species, and the sequences that we retrieve from gene bank from other species of the Ceratopomyidae family. We'll construct 2 trees, one based on maximum likelihood using ecotree, another using Bayesian inference in Mr. Bayes. And then the Bayesian, we will apply some species delimitation methods to determine the molecular boundaries between different species and determine the phylogeny of the family. From this chapter, I'm now doing the DNA extraction, and in further weeks, I will start doing the PCRs. For the third chapter, we would like to determine the ecological factors that determine the species survival, and for that, we will use habitat suitability modeling. For now, for each species, we have the occurrences. Most of them, we're still processing the data from Córdoba, Jaén. And some of the data that we have, we clearly show us that Leptoconops, in case of Leptoconops nigritorax, most of the occurrences are in Huelva, Sevilla, and Cádiz, near the Guadalquivir Basin. In case of Leptoconops irritans, the distribution is more patched. In case of Leptoconops triangularis, most of the occurrences are in Grazalema. And in Leptoconops communis is well distributed through Huelva, Seville, and Cádiz. With all the spatial data that we have, we will clean it, and we will use different climatic and environmental data retrieved from different sources, such as TerraClimate, Copernicus, on Earth data. The variables that we will select will be based on literature of different Ceratopomyidae species, mostly from Culicoides, which will be similar. We'll conduct variable selection and multicollinearity test, and with the variables and our current data, we will conduct species distribution modeling using different algorithms such as random forest, boosted regression tree, or Maxent. 75 of the data will be used for train and 25 for test. We will do individual modeling with a variable, climate variable, and then environmental variables. From each of the individual model, we will choose the variables that have high relative importance, and with that, we will do a combined model with the climate and environmental data. And we will retrieve from the analysis through ensemble modeling the habitat suitability prediction map from each species, the predicted probability of occurrence, and the ecological niche for each species. From this chapter, I'm now doing the spatial data processing, and we'll start in further month the analysis with the habitat suitability models. For the fourth chapter, we want to determine the host preference and detect parasite in the Leptoconops species. For that, we will do 2 analyses. One of the analyses will use engorged specimens. All are sorted individually in Eppendorf tubes by species, date of capture, and site. We will firstly dissect the different specimens into head, thorax, and abdomen, and we will use only the abdomen for this part of the analysis. For the host identification, we will target the COX gene to identify the blood inside the abdomen. We will use COX gene, universal COX-1 gene primers. And for parasite detection, we will use different primers targeting parasites such as Ammoproteus, Trypanosoma, or Nematodefilaria. For the host from the host, to identify the sequences, we will use GenBank, and for parasite, we will use GenBank and Malawi for avian malaria parasite. The second part, The second analysis will be done on unengorged specimens that we have in pools of 15 specimens, all sorted by species, date of capture, and sites. And we will only do a parasite analysis on these specimens. For this chapter, I have the all sorting part, and in further month, I will start the molecular analysis. The fifth chapter is to revise the sandfly species blood feeding patterns, and we will use a systematic review. We conducted a systematic review on Web of Science, searching from the studies about molecular analysis of the blood of different sandfly species in Europe. We retrieved from that search 167 articles, and after exclusion, we included 44 of the articles. We extracted different data, such as species, blood meal analysis of that. And then we will conduct different data analysis. The first analysis that I have is a descriptive analysis of the data from the 44 included studies. We can observe here that most of the blood sources came from mammalian hosts, and most of them from domestic mammals. And that's maybe because most of the study were conducted near animal shelters, farms, or human urban areas. So most of the hosts can be only that. And in case of the number of blood sources from different species, most of the study species is Phlebotomus perniciosus, which is the main vector of leishmaniasis here in Europe. So that's because this may be because being the main vector, the most analyzed species is that. Further analysis will be done on this using generalized linear mixed model. I'm now doing that part, and that's why I haven't here. The final chapter will be to assess the high transmission risk of leishmania in Andalusia. The first part will be to identify morphologically the different sandfly species, to have the different occurrences, and we'll select environmental and climatic data also relevant from the different species. As for Leptoconops, we conduct habitat suitability modeling using the same method. And then we will retrieve the 3 different results, and with the predicted probability occurrence map, we overlap the leishmaniasis incidence rate in Andalusia, and from that we'll construct a risk map of leishmaniasis and detect the areas that have the high risk transmission. From this, I am now doing the morphological identification part, also with the aid of our technical team. Finally, I will talk about my thesis plan timeline. The first chapter is already done and under review. For the second chapter, the 4th and the 6th, I am doing the laboratory work now in different stages. And the 3rd and 5th, I am doing the statistical analysis. The second chapter and the 5th will be finished by the end of the second year, and 3rd, 4th, and 6th will be ended by the 3rd year. The writing of my thesis will take place in the middle of the 4th year. During my research, I will have two stays, one short stay this year and a long stay during the 3rd year. The short stay will take place in the Instituto de Salud Carlos III in Madrid under the supervision of Maribel Jiménez this June. And the long stay will take place in the Pirbright Institute in the United Kingdom. My thesis committee, it's formed of José Martínez de la Puente from Estación Biológica de Doñana and Ricardo Molina from the Instituto de Salud Carlos III. And in my research, I have the aid of some of my collaborators, which are Maria José, Sirin, and Mikel. Finally, I would like to thank my team, and I'd give a special thanks to the technical team, which have done all the fieldwork part and the morphological identification part. And finally, I would like to thank you for your attention. Yeah, I will then.
Thank you very much for presenting your PhD plan. Before we start the discussion round, just a reminder that we have the discussion round in English. So please, questions to be asked in English. Victor.
Thank you very much for your presentation. I want to compliment you on extremely robust integrative taxonomy approach and very ambitious dissertation. First question is really short. Have you talked to Art Borkens?
Yes,
You talked
we
to
have
Art.
talked to a serious time.
And he was supportive.
Yeah.
Great. It's very lovely to hear that Art supports anything done with molecular systematics and taxonomy of ceratopogonids. And can I ask which 3 genes are you sequencing for your third chapter? I'm sorry.
Second, yes. It's in here. This one, the COX-1 gene, we already have, the cytochrome B, and the ITS1, ITS2 region.
You haven't looked at the CAD1 or 2 genes because they show to be very stable markers for phylogenies of colicomorphans. I was involved with some of chironomids and simulate phylogenies, and CAD genes proved to be very useful.
Yes, we are based on one phylogeny of culicoides that have been done.
Yeah.
So we choose that genes. We have to prove it's with work. And that's because we want to do a integrative of the family. So all the genes that we have on GenBank. So
I can only compliment you. It's a very brave task to do a molecular phylogeny for Ceratopogonida, and I wish you amazing success with that. Can you also go to your new species? Just a curiosity.
What?
Yeah, you had a picture with a maxillar pulp. Yeah, so in Triangularis, it's very similar to the fossil genus Eonanthochilea from Cambayamber that was described. I'm not saying it's the same genus, it's Leptoconops to be sure. Do you have any hypothesis why the penultimate pulpomere is the way it is? Because we have published this paper with Frauke Stebner back in the day, and there is this genus Eonanthochilea from Eocene. It has a gigantic penultimate pulpomere, and we did a micro CT on it, and we thought it might be a pheromone reception organ, but of course you cannot check
here
it.
In Leptoconops, as in Culicoide, this maybe is a sensory organ. So this is hypothesized to be for the reception of CO2 detection.
So there is a mix of Sensilla selaconica and Sensilla hetica sitting on the top of it.
We don't know nothing.
All right. Well, maybe SCM it. Okay, thank you again. Very nice presentation.
More questions? Thank you. Yes?
It's only online, the sound.
Okay, thank you very much. It was very nice, very interesting thesis. Why do you think that there are so few records in GBIF of this genera? Because then you show that it's all over in Andalusia.
Yes, that's because it's not studied. We know that they are occurring in Italy, but maybe not in GBIF deposited. But in most of the areas around Europe is not studied this genera.
Is it a native species?
What?
Is it an introduced species? Because
we don't know, but it's not an introduced because they have been recorded from other species like Leptoconops besi and noe here from 119.
Yeah.
So
I'm asking because in Menorca, I think that there was something like they discovered the species recently.
Yeah, they described recently in 2025 the Leptoconops irritans. This one was the first record from Spain of Leptoconops irritans.
Thank you very much.
Do we have more questions?
Thank you. When you said that you are going to look for vectors, for parasites, it's a very general question. What's the normal occurrence? Well, not in this species, but in general. I mean, is the occurrence of the parasites very high, or are there many parasites in each individual, or
On
most of them
leptoconops?
don't wear parasites?
On leptoconops?
Well, in general.
In leptoconops, nothing is known.
Yeah, I know, but
We
in
don't-
general,
But in general, in Culicoides, it's the sister taxon, so I will talk about it. It's known that they can transmit some parasite like Homoproteus or Trypanosoma, but it's not the main or the important vector that can transmit it. So I don't know about the prevalence of incidence of that.
okay, I'll put the question in a different way. How many individuals you think that you have to sample and to find something?
As no other study have done, we will probably do many
But
analysis.
I mean, I don't know what's many.
I don't know,
Thousands?
thousands maybe.
Okay. Thank you.
Do we have more questions from the audience? If this is not the case, thank you very much again
Thank
for
you very much.
your PhD plan. Also, next week we have still an open spot for the seminar. So if one of you has a guest or wants to present, just reach out to us. Thank you.